So, ladies and gentlemen, a massive welcome here to our Biotech Gate Festival, which has been going on. We started yesterday. Is it yesterday, Craig? Started
Correct.
yesterday, and we've been had the privilege and the pleasure of running the Coffee Buddies slot with Biotech Gate since its formation, which was a couple of years ago. And our goal as Coffee Buddies, as all of you know, is to engage, entertain, and inform. And one of the things we really like to do is to get interesting people to come and talk to us and tell us their story and about their company, and often their vision and their dreams. And today is a great opportunity for us to speak to Mikael, who I'm going to introduce now, who is joining us from which part of the world, Mikael, are you in today?
In the south of Sweden, Scandinavia, Europe.
Yes, there's lots of snow. Has it at the moment?
Not really. Sweden is a very long country. It's about 1,600 kilometers tall as a country. So we have snow in the north and bare grounds in the south, where I am.
Oh, fantastic. Well, Mikael, fantastic. Maybe you could introduce yourself and tell us a little bit about your story.
Well, my story. My name is Mikael Lindstam. I'm Swede, and I run companies, started companies almost close to a quarter of a century now, getting closer there. I have a PhD background in chemistry, did quite a long stint within material science, which is my background actually. So what am I doing in biotech? Well, I'm in biotech since 14 years now, slid in on a banana peel in a governmental fund in Sweden that handled a lot of biotech assets. And one of these assets became what you see up there, Aptahem. It's a mirror here. So Aptahem is then a platform company with a few assets in aptamers. This RNA-based aptamers. We founded 2014 out of this foundation where it got the call to close down actually. So we spun out a lot of assets there into companies, or we out-licensed them and so forth. And quite many companies came out of this, but the one I'm representing here as a CEO for this company is Aptahem. So the platform is aptamers, RNA based. So there are kind of a smaller type of antibodies. They're totally synthetically made, which then avoids any kind of endotoxemic or endotox parts from biological manufacturing, which is a big help when you start out with something like we did. These are spawned out of a parasite, the malaria parasite, which actually produces a protein, and this protein glues together red blood cells for its survival, and that leads to something called rosetting, and this rosetting leads to strokes. 100% death from brain malaria. Well, that's the story. So we handle this and look at what can we do for a more market positive development of this asset. And we did. So we found that it could be more than just an anti-rosetting agent. And here we are today looking at it as an anti or a modulating thrombin modulator. And it also only goes on one part of thrombin. And
so maybe Mike, you could just give us something about the landscape.
Okay, the landscape. Sure.
So if you take, the world of, you say it's an antibody-like molecule.
Yep.
I mean, the world of biologics has exploded really. So you've got you've got mAbs, you've got bispecific, trispecific, you've got T-cell engagers. Where first of all does this sort of aptamer type of technology fit in? Maybe you could help us with that first.
Well, it's an antibody. I see it's a replacement for antibodies in many ways, because you can actually manufacture it in a better way. So it is a synthetic biologic in a way, if you look at it. And these RNA fragments, how you build these aptamers, can be very good fitted to different targets. So I would say that we are a smaller, more efficient to specific targets, and targets not that no one have been able to target before. You can do this with this RNA technology, and that is an oligonucleotide, by the way. So you just play with Lego pieces, which are the RNA pieces, to put a perfect fit to some kind of a target. So you get very good fit, very good stability, and very good efficacy to the target.
And can you do sort of like we do with antibodies, bring things close together, sort of different immune cells? Can you bring them together, or is it really the Aptamer is more focused on I don't know, like a small molecule, better than a small molecule, but a small molecule fit to a target?
Yeah, they're not that small, but they're very
smallish.
formidable. Well, let's say our Aptamer is about 2 nanometers in size, so that makes it a bit more interesting than an antibody in that purpose to reach hard to get targets. But I would say that I mean, the technology is quite new, but it's been around for a long time, because
yes,
the challenges have been manufacturing these RNA aptamers, not so much getting smart aptamers. And you use a process called the Celix process, which was an invention use patent, where you have a lot of fragments, like say a big swimming pool, and they swim a lot of RNA fragments of different stature, and then on the side walls of this basin sits the target, and it's a driven process to optimize on a target. And then the Celix, it says itself, it selects those who have the best affinity to these targets. So out of a catalog of I begin 80,000 down to 10,000 down to 500 down to 10, and then you test them, and then you see that there are 3 which we have in protection to this target that had the best efficacy and adherence.
So what was your process that you went through to identify the target that you are going after?
Well, that go back to the story actually with the protein created by the malaria parasite actually, but it's a very big protein and had a lot of active sites. And while doing the Celix process for this protein, we are not really sure, but there's many sites, and one of the sites seems to be the heparin binding site, which is part of the thrombin, actually. So that's we believe is the most crucial bind site of that protein, and we see that also when we do our mechanistic studies here, that it binds very exclusively to exosite 2, which is the heparin binding site on thrombin, which I were talking about in the beginning here, and it does so without binding to any other sites on the thrombin, which then avoids side effects, which is usually associated to the antithrombotic drugs like heparin in this case, which also gives a bleeding effect, which we do not see with this drug candidate.
So this is an antithrombotic.
Correct, that is. But this works in the coagulation sphere, but it's not an anticoagulant. We can show that it's not an anticoagulant. It's strictly an antithrombotic, just like you say.
And what's the market like for antithrombotics now?
You beat me on that one. Depends on how you define what we want to do with our drug, of course. We are targeting sepsis, and we will for sure target something more specific down the road in clinic now, but we're just entering clinic 1, so that's the first steps to define that. But we see it as an antisepsis drug foremost, and not as an antithrombotic. That describes the mode of action. But
what's the challenges, Michael, of developing, or not developing, but of bringing a new antisepsis drug to the market? What's the sort of challenges?
Yeah, I think most of you know it's a huge challenge. It's the biggest graveyard in development.
Well, the pancreatic cancer, I think, is it pancreatic?
Well, there's another one, of course, but sepsis is associated with so many deaths globally. I mean, I would say it's one of the biggest killers we have, and that's because you not always associate deaths to sepsis, but COVID, most of them sepsis, cancer patients, low immune defense, get sepsis in the end. So all these things play a role, but when you talk about this, you say that person had pancreatic cancer and blah blah blah, etc. So you miss actually the final point sometimes, where people actually die.
Yeah.
So it's a big iceberg, and you just see a small top of this. And
I mean, I'm my background was originally pathology, and what I've seen over the last few years is that diseases are getting subdivided left, right, and center. I mean, is sepsis the same? Is its definition and its specificity changing?
Well, let me break it down a little then. I mean, we see that 2/3 of all sepsis septic cases are bacterial. And you give, in standard of care, you give antibiotics, but the rest, 1/3 is viral, fungi, or it could be even non-pathogenic, like SEERS, for example. And we have all these examples of trauma to the body and other things that happen. But if you're going to be more specific, you have DIC, you have renal failures that occur at shock and so forth. You have all the tissue breakdown, hemorrhage in lungs and so forth. So there's many things that can happen during a septic event, especially when it becomes severe or when you turn into shock, of course, when the system starts to leak and you get hemorrhages. So you know, it's very tough to diagnose, actually, when you come into the emergency care. So a companion tool would be fantastic for us that developing this, and we are looking at such as we speak right now that could be very quick and to the point, actually, and there may be some AI involved in this as well. But the future will show us where that leads. But anyway, it's a very tough, I call it a syndrome or condition. It's not a sickness because it compromises several diseases, actually, or sickness syndromes or issues when people get this. So it can turn anyway. And if you're lucky, if I say that bluntly, that you may get away with amputation or just a kidney transplant or something like that. And we know that it goes very, very quick when you slide into shock. And today, where you slide into shock, there is absolutely no treatments, more or less. The doctors sit by the side and wait for someone to die, which is very, very sad. And incidence is more prone on older age groups as well as infants or neonatals. There's the biggest death rates, actually. If you have a good immune system as a grown-up, you can handle somewhat a septic condition, but you need to get care as soon as possible. But I could talk all the way about this.
Well, no, I mean, it's interesting for people. Will it be that, I mean, I'm not thinking of a pure prophylactic, but would your drug be given early in the treatment? You know, when you're treating with antibiotics, will you be giving this drug early, or is it when you're actually starting to get the full-blown septic shock?
Well, there's the challenge with standard of care. You know, it has to fit in with other drugs given or treatments during that. But I would say more when, excuse the expression, the shit hits the wall with the patient, there it could have a big impact, as we see it from the studies we have performed. And it's not the prophylactic. I mean, who knows, there will be septic or not, you know. So, but it is a very fast acting drug. It's an IV solution. It's totally made for critical care, and it takes, you know, 10-15 minutes, and it's doing its work after an injection. So it's very efficient, and it's really definitely.
You can't load up someone with cytokine drug so prophylactically, so that eventually, if there's a storm, they get treated. You can't. Maybe that's a mad idea.
No, I mean, they could, but, you know, still they are quite strict with standard of care procedures, you know, and the first responder is IV antibiotics when you come in and show signs of sepsis. You know, and also these other treatments like anticytokine treatments and dexamethasones or whatever they use, and anticoagulants. I mean, they all have side effects. Either you bleed, or you don't have effect enough, or you have a suppression of the cytokine storm, but also the side effects of suppressing the system that actually works may even create more havoc with the patient. And we can see that our candidate, which is called Aptahem, does not suppress the immune system, actually tickles it and you know, helps it up again on its knees. So it's a modulating kind of a drug candidate. We don't suppress the system totally, just enough of the body to take charge again. That's what happens.
So just thinking of the development path, where are you on the development path?
Well, right now, as I mentioned, maybe in an introduction, that we are right now getting into phase 1 studies. And we decided to divide it in 2 parts. Of course, the mandatory healthy individuals to check for safety. And then we have a B part where we do a challenge study with liposaccharides. And we have used that throughout our preclinical studies as well. And that's to elevate the inflammatory response in the body. And then we see what our drug can do. So we have a readout on some crucial biomarkers for the critical care clinic. And the nice thing with and we were very proud of that, and to our knowledge, we are the first ones, or among the first ones, to give the drug therapeutically. Otherwise, usually when you do this kind of studies, provocation studies and so forth, you have to give it what do you say now, prophylactic.
Yeah.
And so we're very proud of that, because that's from our plus 6,000 pages of talks and safety studies that show very, very safe drug. So we're doing this study we wanted kind of an early proof of concept, and also to do risk phase 2 studies that are upcoming. 23, 24. So now we have 23 to do the first in man and the provocation study. So It's very interesting. It's really exciting times for us to do this, and we have a lot of data through the years, preclinical studies, mechanistic studies, and they will now soon, I hope, be published if editors and others see the elevating information that we want to show the world there. So it's a lot of things happening right now.
So have you got a big team, Mikael, or are you a small team? Or?
Big team, small team, depending on what continent you are, I guess. But we are a semi-virtual company, becoming more a normal company. But let's say people that are activated in the company is about 50-50 consultants and employees, and at times we sometimes are 20 people active, not full-time, part-time consultants here and there. We have some full-time consultants that work for us in specific areas, but we are a very tight team. So we see ourselves as a think tank management team doing things out of the box, and then we buy all the services, but we manage them and we tailor them, because standard setups for different CROs and service providers will not do a drug justice that the one we have, for sure. And we put a lot of pride and honor in what we have done so far. And
do you have a portfolio with other things in it?
Well, the portfolio are the 3 Aptamers, which we call the platform. So we still have some more to work on. And now when we are in clinic with this first one, our lead candidate, we are, of course, eyeing at a second pipeline. And we already did a few small tested on what we call Apta2, which is spawned out of the same target, the original target, but they structurally look totally different. So you can work with these and find new interesting indications to work on.
Interesting. And so over the next three years or so, Mikael, what's going to be, how's things going to progress, do you think?
3 years, in the best cases, we have done a deal of the proof of concept in man. That's what we are aiming at. Of course, the sooner the better, but if you want to make a good deal and it's attractive for the potential partners, then it's a bit more money involved, of course, and good for the shareholders that I represent every day. And that's our goal, of course, and it's no lie to say that usually there it happens, but I'm trying to find elevating points and trigger points along the road and milestones for the company to share and to elevate ourselves to the attraction of someone's interest.
And I mean, your purpose of coming on the show tonight is to really engage people in your story. I mean, are there sort of 3 things that you'd like them to remember of what you're talking about?
Well, Aptamers may have a future. Sepsis is an awful condition, which many people do not know too much about, actually. And I do believe that Aptahem have a solution to be a big player in this field, as long as it holds up in the clinic, which we strongly believe it will.
And that is a lovely, that's a lovely segue, my friend, into us asking some questions. So I'm going to open the floor up. I can see I've got a couple in the chat room already. But I think
Tom's got a question. Yeah,
I think that I've got 2 in the chat room. So let's, Tom, maybe you could start.
So, as I said earlier, I think it's a fascinating technology, and I've really got a comment and a couple of questions, but let's start with the comment. why do you think there's been such a lag time in the success or seeing success with this platform approach? Because I think there's only one FDA approved drug, you know, which is for from Macugen, isn't it? The Pfizer iTech, you know, collaboration. And as I mentioned earlier, you know, one of my old CEOs who started a company in this area back in 2006. So it continually surprises me why this technology hasn't advanced so quickly. So that's the comment. The question regarding your compound is, how are you differentiating that? Say a compound that is already out there, which is active. I think that's the NU171 compound. So, you know, there's competition there, and I thought that your particular compound may have other properties to differentiate it. Is that correct?
Yeah, we'll call it disruptive, I have to be frank. Thank you very much. Very good questions there. Well, the technology Macugen, it's actually withdrawn now, and it was quite painful, you know, injecting the eye for the fine vascular there. I think so. In the beginning, it's been tough to prove its potential due to, as I mentioned during my talk here, is that manufacturing of these aptamers have been tough. And also aptamers are at least are RNA based, but they're also DNA based. But you know what happens when it comes into the body? They will be very fast consumed. So we have fluorinated our aptamer, to say, so it's a Teflon aptamer in that case, and that keeps it alive much longer than it would otherwise not being attached with some kind of. But you know, Macugen was attached to PEG, and you know, PEG in the human body, it's not a hit, I would say so. So that's not a good one. I think so that the inherent issues with aptamers in this case is RNA based, is that they have short lifespan, and because and today there are better life extenders, you know, that's lipos and whatever you can as a delivery tools in the body, and also manufacturing that I was talking about. It has some challenges to make big batches. And the longer an aptamer is, the theoretically the less pure it will be. Yeah, that's just how it works when you do this. And also, so I think so, having breakthroughs in manufacturing and more people engaging into aptamers, because when I'm on the field, I hear this pharma, that pharma. Japan is very big on aptamers, or RNA-based aptamers, DNA. And there is some success cases coming up now, and I hope they will hold all the way to the market, of course. But it has taken a long time to get traction, I think so. And the proof in the pudding is, of course, something on the market that shows its superiority to other drug types. That goes without saying. So we just have to keep on fighting and deliver good results. But I would say so the inherent qualities of these are that they are non-toxic and very safe. And they can be very direct to the target with high efficacy and so forth, and actually hit targets that you wouldn't be able to hit otherwise. So there is a potential in this, but we need more success stories for sure. And I hope to be one of them with Apta1 and Aptahem, of course.
Fantastic. We've got another question from Katrina. Katrina, would you like to come off mute and ask a question?
Sure. Thanks, Michael. That was a great discussion. So I think you answered my question. Just to clarify, you're a chemistry guy. So is the manufacture of your aptamer right now a chemistry or a biological process?
That's totally synthetic chemistry. Yeah.
Okay.
Yeah.
Yeah, and that I would say that's it ought to provide enough of a platform for other synthetic chemists to work on improving the approach, right? That long-term manufacturing approach. I'm assuming also that again, sepsis is a great opportunity because typically those administrations are done IV, so you're not really having to worry so much about creating an oral form of the drug. Correct. Are you planning to test? So I think one of the challenges finding a sepsis treatment is really the identification or the diagnosis of sepsis. I think that's a big challenge.
Yes.
So the question, if they are really safe and effective, I think the part of the target would be to test a very early administration protocol, sort of a CYA protocol as an arm. Are you planning to do that in your clinical trials if you don't get an acquisition at the end of 1B?
Absolutely correct. Thank you. Very, very good questions there, and well pointed. Yes, that is the case. Of course, we would seek any kind of a fast track or even orphan if that was possible. Anything to accelerate towards the market. I mean, we are backing our comments with a massive amount of preclinical studies in different types of organisms, and it's not only rodents. We have also been in the NHPs, which is crucial to make the proof for such a drug candidate and for such a field. I mean, when we saw the early results from preclinical studies, we realized that we're sitting on a gem here, and would it hold? And so far it did. You could really reduce the death rates on anything, and you could suppress the cytokine storm. You could have an antithrombotic effect, so there was no bleeding, there was no thrombosis. We could even see that you can reduce thrombosis with this candidate. So it does a lot of things. It's like a multi-mechanistic candidate, and we know more today that it does all these things through just hitting thrombin on very specific place, like exercise 2, the heparin binding site. And by that, you also get a very rapid response from an anti-inflammatory and anti-thrombotic response by just stopping the platelets having their way with the inflammation.
It's fascinating that it's still so effective, even though you covered it with fluorines.
Yes, I mean, yeah, because it has to do with that the fluorine is sitting on what is it, guanines or something like that, and so it doesn't interfere with, because it's a folding structure. It's always foldings to keep it stable, and of course it has a geometric docking where it does its exchange when it hits the target. So it's really not in the way. It just keeps the structure alive a little longer from the enzymatic attack in the body.
So I think we got time just for one more quick question before we go into breakout. So Anna, you had a question?
And also Keith had a comment.
Okay. Anna, if you'd like to go first with your question.
I mean, from the international markets, I mean, there's
Can you speak up, Anna?
Can you hear me?
It's not great.
Can you hear me
that's
now?
better.
That's
okay.
Better. Yeah. So Mike, thanks very much. So I think there's lots of unmet need also in the international markets, but you're in very early stage. But I think also sepsis might be also military, defense or military on the field. That one is another area of unmet need. But in terms of international markets, have you identified any, can I say Japan or something? Have you identified anyone out there, target, like a target international market, which means outside Europe or US?
Yeah, I mean, we target globally, to be honest with you, going back to the military, yes, that is a hot lead, and we are actually members of the US MTech Association, which have the military and the government involved there. It would be nice to have some DARPA contacts as well, but and sepsis is a big problem with wounded soldiers in the field. So, and also we have the malaria part. I should not forget that, but now we are focused on sepsis, of course. I didn't really get your last question there, Anna. Could you repeat that, please?
No, my last question, apart from the military, is about because it's an unmet need and there is no effective. I think within the international markets, because I mean I work on the international strategy. I have been working. There's lots of like very high value and lots of growth there. Or as I said, through unmet need, you can have fast track. In terms of your registrations. But if you need anything in terms of international strategy, always happy to speak offline.
I'm happy
Very
to
good.
connect.
I appreciate that. Thank you much, Anna.
Absolutely. Happy to connect. And a final comment, I think, from Keith before we go into breakout rooms. So hi, Keith. Great to see you.
Hello, everyone. Yeah, hello, Mikael. I missed the beginning. Sorry, I was trying to get in, had a problem. So sepsis, a massive unmet medical need, so a really good target to go for.
Yes.
Really wish you luck. I know several people One actually died of it. One I saw in A&E being resuscitated and unfortunately came through.
Yeah.
I don't think hospitals fully understand sepsis, you know, and care homes and all those sort of areas, even though some of them have the guidelines splattered all over the wall, sort of saying what to look for in sepsis. I just don't think it's trained early enough. And so from your perspective, it's at what point are you going to target? And I think somebody raised it. How are you going to select those patients where you can get that benefit?
Well, we know, thank you. A very good question, and as we're working constantly with these questions, and hence we also will engage in this provocation study to give us a little guidance, at least to begin with. And I think it will be very resourceful for us to get that study done, and as I mentioned, also to de-risk phase 2, and then we come to the cohorts or the selection there. It's not an easy one, and you know. We are not really there yet, but we think we will be in an area where severity sets in, as we can see from the character of drug. But it's a little too early to comment on that, even if we're looking in that right now, and we are, as we speak, reformer or TPP as well. But we have to stay calm a little bit more to see where we land. But I think so. The hardest part of a proof of concept study will be the selection of patients and how they're chosen, of course.
Because the difficulty is if you choose too early, then people, it'd be quite hard to get there. If you choose too late, you might not, even if it's a successful drug, it might not work.
No.
The constant challenge. Graham, thank you, Keith. Let's move into the next phase.
Yeah, absolutely. I just want to thank Michael for.
Let's have a big thank you for Michael. We
really interesting. Thank you.
Maybe you come back.
Appreciate it
another time and give us an update.